these pictures, what do all these foods which many of us consume every day have in common? So if you guessed starch, then you are correct. But if you did not, then it's alright, because that is our topic for today. It's all about starch. But before we delve into the topic, let's first state the learning outcomes. First, we'll define starch and explain its biological significance. Second, identify the two primary components of starch, which is amylose and amylopectin. And lastly, differentiate the two primary components of starch in terms of structure and function. So what exactly is starch, and why is it so important? So connecting from the previous lessons about carbohydrates and polysaccharides, starch is one of the examples. According to Britannica 2025, it is a white granular organic chemical that all green plants produce, with a molecular formula of C6H10O5. It is a polysaccharide that has glucose monomers joined in an alpha 1 to 4 linkages. As stated by Cleveland Clinic, it is commonly found in foods like rice, potatoes, corn, wheat, like the picture shown before, and so much more. In short, starch is nature's way of storing energy, and when we eat it, it becomes our energy. So quick pit stop before we move on to the next topic. So the question is, what linkage does starch have? If you answered A, then you are correct. If not, don't stress. Mistakes are part of the process. Understanding comes with practice. So now, let's dig deep into how exactly starch becomes our energy. Converting starch into energy is actually done through a series of reactions. We call this digestion and absorption. So first is digestion. When we think about digestion, the most common misconception is it first occurs on our stomach. But in reality, it actually starts when we chew food in our mouth. We call this partial digestion. There is an enzyme, a biological catalyst in our mouth called salivary amylase, or ptyalin, that initiates the digestion of starch by breaking it down into smaller sugar units. This will travel down into the stomach where it slows down the digestion process because of the acidic environment caused by the stomach acid, HCl, deactivating the enzyme. Once it travels to the small intestine, the process continues, and with the help of another enzyme called pancreatic amylase, where it further breaks down starch into maltose. This maltose will be broken down by maltase, another enzyme in the small intestine, into glucose. In this diagram, it can be seen how, with the help of amylase, starch is broken down into maltose, a disaccharide. Then, that maltose is broken down by maltase into glucose, a monosaccharide. So, from a polysaccharide, starch, with the enzymes such as amylase, pancreatic amylase, and maltase, it was broken down into a monosaccharide, glucose, the simplest form of carbohydrate. This means that the final product of the hydrolysis of starch is glucose. This glucose is then absorbed in the bloodstream where it is transported into cells. Once in the cell, it enters a series of biochemical reactions called cellular respiration, producing ATP, the energy currency of our body. So from this diagram, we can see how from carbohydrates like starch, it is broken down into glucose. This glucose undergoes glycolysis where it produces pyruvate and ATP. This pyruvate undergoes pyruvate decarboxylation where it is converted into acetyl-CoA. The acetyl-CoA then undergoes Krebs cycle where it produces 3 NADH, 1 FADH, 1 ATP or GTP depending on the cell, and 2 carbon dioxides as waste. This then undergoes the electron transport chain, producing ATP. This ATP is then used all throughout the body. It powers everything, from muscle movement to thinking and even cell repair. Think of ATP as your phone battery. A phone needs a battery to function. Your cells need ATP to perform any task. When the ATP or battery runs low, the cell must recharge it by producing more ATP through respiration. And we can help ourselves by eating foods with the right nutrients such as starch. And that is why starch is so important, especially in its role in energy. Without starch and the glucose it provides, your body would struggle to keep up with daily activities. So let's have a quick pit stop before we move on. So the question is, what is the enzyme used to break down maltose to glucose? If you answered B, you are correct. If not, that's totally okay. Now you know. Let's keep learning. Now, while we talk about starch as one thing, it's actually made up of two distinct components, two different types of glucose chains, each with its own unique personality. These two components are amylose and amylopectin. First up, we have amylose. Let's call it the straightforward one. Imagine a long straight chain of glucose units all lined up neatly. That's pretty much what amylose looks like. So the basic structure of amylose is made up of thousands of individual D-glucose sugar molecules, and these glucose monomers are linked together in a specific way by an alpha 1 to 4 glycosidic bonds. Think of it like this. The carbon atom at position 1 on one glucose molecule forms a bond within the carbon atom at position 4 on the next glucose molecule. And because it's an alpha bond, it has a specific orientation that allows for the famous coiling. This linear arrangement is what allows the chain to coil into a beautiful helical structure, almost like a spring. This compact shape is super clever because it allows plants to store a whole lot of glucose in a really small space. Fun fact and a cool test. When iodine is added to amylose, it fits neatly within this helix, producing a characteristic blue-black color. This is the basis for the classic iodine test for starch you might do in a lab. Now, what's its job? Well, because it's a relatively simple straight chain, our bodies find it a bit harder to break down compared to its counterpart. This means it tends to slow down the release of glucose into our bloodstream. You can think of amylose as a steady, slow-burning fuel for your body. When you cook foods high in amylose, they can sometimes feel a bit drier or firmer. This is because its linear structure makes it less soluble in water and gives it a more rigid texture. It also contributes to something called retrogradation. That's when starch crystallizes and becomes less digestible upon cooling. Ever notice how bread can get stale and hard the next day? That's amylose at work. Pit stop! Alright everyone, let's take a quick pit stop here. Thinking about what we just learned about amylose's structure and function. Which of these foods do you think would be relatively high in amylose? So the correct answer is C. If you got it right, great job! You're on the right track. If not, no worries. That's why we're here. To learn, not to be perfect. So let's keep going. Next, let's meet amylopectin. We call this one the bushy one. If amylose was straight line, amylopectin is just like a highly branched tree. Just look at all those branches sprouting off the main chain. This super branched structure is key to amylopectin's function. The basic structure of amylopectin consists primarily of glucose units linked by an alpha 1 to 4 glycosidic bonds, just like amylose. However, here's the crucial difference. It also has numerous branch points formed by an alpha 1 to 4 glycosidic bonds. Imagine a long main chain with an alpha 1 to 4 glycosidic bonds. Then, every roughly 24 to 30 glucose units along that main chain, a new branch sprouts off. This branch is connected by an alpha 1 to 4 glycosidic bonds, meaning the carbon 1 of the new branch connects to the carbon 6 of the glucose on the main chain. This extensive branching creates that compact, tree-like structure. Because it has so many ends available, our digestive enzymes can get to work on it much more easily and rapidly than with amylose. This means amylopectin provides a quick burst of glucose. Think of it as an immediate energy source. The highly branched structure of amylopectin also makes it more soluble in water. This is why when you cook rice or pasta, it becomes wonderfully soft and sometimes even sticky. That's the amylopectin doing its job. So let's put it all together. So amylose has a long and linear structure, and its function is for slow digestion. And we have amylopectin, so its structure is highly branched, and its function is for fast digestion. So how does this differentiate them in terms of their role? Amylose's structure, which is linear and coiled, makes it less soluble in water and gives it a more rigid structure. This is why some starchy foods high in amylose can feel a bit drier or firmer. It contributes to the retrogradation of starch, meaning it can crystallize and become less digestible upon cooling. Think of it as stale bread. So amylopectin's highly branched structure makes it more soluble and allows it to form thicker, gel-like structure when heated with water. This is why when you cook rice or pasta, it becomes soft and sometimes even sticky. It's the amylopectin at work. Now let's have another pit stop. So the question is, this is a component of a starch where its chain is a long straight chain of glucose units. It is sometimes called the straightforward one. So the correct answer is B. If you got it right, great job. You're on the right track. If not, no worries. That's why we're here, to learn and not to be perfect. So let's keep going. So in summary, starch is a complex carbohydrate that plays a vital role in energy storage and supply, especially in plant-based foods. During digestion, it is broken down into glucose, which fuels the body through ATP production. Starch is made up of two components, which is amylose and amylopectin. While both are chains of glucose, amylose is mostly linear and the straightforward one, whereas amylopectin is highly branched and the bushy one. Understanding their structure helps us see how efficiently the body can access and use this energy. Now that we have discussed the definition of starch and its role in energy, as well as its components, which is amylose and amylopectin, and their structures, let's have an assessment to check your understanding of what we've covered. Match the statement of each number to its corresponding component of starch according to its structure. Good luck!